increasing bioluminescence in plants.
So what exactly does this mean? In labs, scientists have been able to produce bioluminescent plants by inserting a series of genes called the Lux operon into the genome of tobacco plants. These plants are then able to emit a yellow light with a greenish tint because of the color of the leaves. Our project is going to be seeing if we can increase the amount of light that these bioluminescent plants are capable of emitting. And we will do this using microbombardment techniques and vector DNA, which we'll explain in a bit.
So bioluminescence is a multifunctional organismal property that's existed for thousands and thousands of years. In essence, it allows organisms to glow. Today, we'll be focusing on one organism in particular, shown on the screen, Photobacterium wagnapi. In Photobacterium, bioluminescence is controlled by 6 genes, called the Lux operon. These genes produce a protein called luciferase, which, when it reacts with its substrate, luciferin, allows the bacteria to luminesce. With the advent of transgenes in the 1980s, scientists became increasingly interested in getting these bioluminescence genes into foreign organisms. However, there were a whole host of problems that came with that, which Wesley will explain right now.
In previous studies, scientists have been able to produce bioluminescent plants. However, these plants are not autoluminescent, which means that they have the genes to express the luciferase enzyme, but they lack the substrate luciferin to act upon in order for the plants to bioluminesce. To remedy this, the scientists would manually add the substrate into these plants, but this could be a tedious process. So in a later study at the State University of New York Stony Brook, an autoluminescent plant was produced by the insertion of the Lux operon into the chloroplast genome. It should be noted that in these previous studies, there has been no indication of a decrease in vitality as a result of the bioluminescent genes being expressed. Moreover, there has also been no indication that the bioluminescent light being emitted from these plants have decreased over time. Finally, the autoluminescent light being emitted from these autoluminescent plants is fairly dim. So in our study, we're looking at how we can increase the amount of light being emitted by these plants.
So like Wesley just said, the goal of our study is to not create bioluminescent plants, but increase the amount of light that bioluminescent plants can emit. And this has never been done before. We'll do this by altering the genome of the plants that we're testing in 2 ways. The first is by inserting something called Scheinfeld-Garner sequences into the genome of the plant we're testing. These sequences are strands of DNA that initiate protein synthesis. The idea is that we will increase the rate of the proteins are made that cause bioluminescence, thus increasing the amount of light that the plant can emit. The second way is by inserting something called RIBA sequences. These are also strands of DNA that code for the production of a protein called riboflavin. So why do we care about riboflavin? As Allison mentioned before, bioluminescence works when a luciferase enzyme acts on a substrate called luciferin. Riboflavin is one of the many proteins that makes up this substrate. When enzymes act on a substrate, they will do so until that substrate runs out, or there's none left. So the idea is that by increasing the amount of riboflavin, we increase the amount of substrate, which will increase the amount of work that the luciferase enzyme can do, increasing the amount of light that the plant is capable of emitting. The long-term goal of our project is to test our methods of enhancing bioluminescence in other species of plants. Like I mentioned at the beginning of the project, at the presentation, what's commonly used in labs is the tobacco plant because it grows quickly and is relatively durable, so it makes it easy for scientists to use it. We'll be starting with plants that are similar to the tobacco plant in size and structure, and working our way up to larger plants, maybe even trees, to see if we can replicate our results.
So now you may be wondering, is our project even feasible? And yes, it is. Scientists have successfully achieved bioluminescence in tobacco plants. The only issue was that the light produced from these plants was not bright enough for it to be used practically. That's where our originality comes into play. Our goal is to increase light production so that we can use it in future applications. Millions of tons of carbon are emitted each year just from artificial lighting. So if we were able to achieve our goal, then we'd be able to reduce that number greatly. But like we said, this is our very long-term goal. Furthermore, there'd be no additional harm added to the environment because we're using mainly organic materials.
So by now you're probably wondering, how exactly do we get these bioluminescence genes into the plant itself? So this is a 2-step process, the first of which is vector construction. When I say vector construction, that sounds super complicated, but all it means is that we're essentially copying and pasting different sections of DNA together that are going to help us get these genes into the plant and express them effectively. So the first thing we're going to need to get in our plasmid vector is something called the promoter. This just allows transcription to begin, which can then start the process of making our protein luciferase and our substrate luciferin. The second thing we're going to get in our plasmid vector is our Scheinfeld-Garner sequences. So this will be added just upstream of where we're going to put the RIBA gene, which I'll talk about in just a sec. but these Shine-Delgado sequences are going to increase our rate of translation, therefore increasing the amount of protein that can be made, increasing the number of reactions, and therefore increasing bioluminescence. The third thing that we're going to add is our Rib A gene. Keep in mind this makes riboflavin, as Sophie said, which is a component of the luciferin substrate. When we add more luciferin substrate, we can have more reactions and thus more bioluminescence. And the fourth thing we're going to add is our selectable marker gene, usually an antibiotic resistance gene. This just allows us to ensure that everything has been incorporated correctly and can be moved into the genome of the chloroplast within the plant. The second step in our process is microbombardment techniques. So we'll be coating small particles of gold or tungsten in these newly formed plasmid vectors. We'll be loading them into a device called the gene gun and essentially shooting this plasmid vector into young plant tissue. And it's as easy as that to get the new genes incorporated into the genome.
So how are we going to actually measure the amount of light produced from these plants? We're going to be using a device called a photomultiplier, and what this device does basically amplifies the amount of light produced so we can actually measure it. How it works is that the photons from the light will hit the photocathode plate, and the energy from these photons will transfer into the electrons and shoot them off that plate. The electrons will then bounce throughout the chamber and hit the diodes, and as they do that, the amount of electrons will amplify until they all reach the anode. That current output will be recorded on a computer, and so basically how this works is as the light intensity goes up, so should the current output. This seems fairly complicated, but it's actually super simple. All you do is shine a light source on the device and look at your computer for the number.
So, our main expected outcome is any increase in bioluminescent activity above the levels already seen in previous studies. As Sabrina mentioned, we'll be using a photomultiplier to measure the amount of light emitted by the bioluminescent plants produced. In the initial experiment that produced an bioluminescent plant, the plant emitted 82 times 10 to the 6 photons per minute. This will be our basal level, which means that if we can produce a bioluminescent plant that emits more than 82 times 10 to the 6 photons per minute, we're on the right track. Moreover, we're hoping to generate new knowledge on how to regulate the expression of genes in these plants. And finally, we're hoping to be able to replicate this method in other plants with various leaves or other desired traits for future use.
One of the problems that may arise in our transgenic plants is that the increase in light intensity could be so minimal that we would have to alter different parts of the plant genome in order to increase bioluminescent properties. Our method might also not be replicable in larger, more complex plants, such as trees, which would limit our long-term implementation of the project in different environments. This long-term application will also lead to interactions with the ecosystem, such as cross-pollination. Cross-pollination is a problem that can be solved by using sterile plants which lack pollen and seeds and thus cannot reproduce. We will also try to limit interactions of our plant with animals by using plant species that are not part of their diet, such as the poisonous tobacco plant. Our long-term goals may be fairly distant. However, we will receive more immediate results for a question of intensifying light, since the tobacco plant is one that grows fairly quickly. Our cost of experimentation can also run fairly high. However, reducing carbon emissions and saving the planet is a goal that is priceless. This leads into our future applications, which again are fairly long-term. Artificial lighting is a really big problem worldwide, and in the United States, street lighting alone accounts for almost as much carbon emissions as 3 million cars and is an $8 billion industry. We hope that with advances in our project in bioluminescence, we will be able to reduce the amount of indoor and outdoor artificial lighting that is used in urban environments. Although the plants will always be emitting bioluminescent light, much like our glow sticks, you simply won't be able to see them during the daytime. It may not be an easy task, but together we hope to make a brighter future.